Object recognition is a very important process that works with other visual and perceptual processes in our brain to help navigate the world around us. While much is still unknown about object recognition, we are starting to find out more about the brain pathways and pathology that contribute to the process and neurological disorders affecting object recognition. One major condition we will focus on is agnosia, and more specifically, prosopagnosia. To understand these conditions, we will first look at the visual pathways involved in object recognition. Much of our visual information first goes to the primary visual cortex, also called V1, which then carries that information to regions of the parietal and temporal cortex. These regions are separated into two different pathways: the ventral stream, which enters into the temporal lobe, and the dorsal stream, which enters into the posterior temporal lobe. The separation of visual information into these two streams led scientists to suggest that one provided what the information was and one provided where the information was. Through many studies, they found that the ventral stream is most likely specialized for object perception and recognition, and therefore is the what pathway, and the dorsal stream is most likely specialized for spatial perception, and therefore is the where pathway. Although separated, these two streams communicate and work together to aid in object recognition and other visual processes. Studies looking at the neuroactivity in both of these streams further supports the idea of a what and where pathway. For example, the receptive fields of the neurons in the ventral stream, the what pathway, always include the fovea, which is used for central vision. This makes sense for a pathway controlling object recognition, as we usually look directly at objects when identifying them, so we would take more advantage of the acuity of the fovea. Cells in the ventral stream also have a high level of selectivity for objects. Early in processing in the posterior region of the temporal lobe, cells are selective for simple visual features like edges. Further along in processing and the ventral stream, cells are more active for more complex features like flowers, apples, and body parts like hands, as seen in this single cell recording to the right. This can aid in object recognition and recognition of very specific objects.
With respect to deficits in visual object recognition, visual agnosia is the inability to recognize objects in the visual modality that can't be explained by other cognitive deficits. Individuals with visual agnosia have allowed for further understanding into the processes that underlie object recognition. Generally, patients with visual agnosia have difficulty recognizing objects that require visual representations. Deficits are solely restricted to the visual domain, making visual agnosia modality specific. In regards to visual agnosia, there are three major subtypes of visual agnosia. They include aperceptive, integrative, and associative agnosia. In individuals with aperceptive visual agnosia, developing a coherent percept is the recognition problem. The unusual view as object test is one way to examine this type of impairment. With this test, participants are shown two photographs of an object, each from a distinct view. In one photo, the object is placed as standard view, and in the other, it is depicted in an unusual view. Individuals with aperceptive agnosia can name the objects in the prototypical orientation, but not the objects in unusual orientations. In aperceptive agnosia, rudimentary visual processing is still intact, but individuals have little or no ability to discriminate between shapes. This is regardless of whether they are faces, letters, or objects. And these individuals also have no ability to match or mimic basic and simple shapes. Integrative visual agnosia is a subtype of aperceptive visual agnosia. Individuals with this type of agnosia can perceive the parts of an object, but are unable to integrate them into a coherent whole. Essentially, patients can perceive local objects correctly, but can't process them completely as a whole. Lastly, in associative visual agnosia, perception occurs without recognition. Individuals with associative visual agnosia retain the ability to perform perceptual groupings, but they'll be unable to draw the same picture from memory, although they do not have general memory problems. In summary, individuals with aperceptive agnosia can process crude visual information. However, they lack the ability to derive the more complex visual information that is necessary to take away shape information. On the other hand, patients with associative agnosia can perceive much more detailed information than those with aperceptive agnosia. However, the subtle perceptual deficits in associative agnosia imply that it might be too simple or basic to conclude that these patients have intact perception.
Though several studies have been conducted over the years to better understand the many mechanisms that go into the process of object recognition, there is still much to know about the field. A study done by Greg Reynolds focuses on infants' development of object recognition. The study as a whole focuses on several mechanisms, such as attention, encoding, and recognition of visual stimuli, and how they all play a role in the development of object recognition. Specifically, the researchers plan to observe the significance of processing non-repeated and repeated stimuli on infants through behavioral, psychophysiological, and neural measures. In order to do so, the researchers plan to place the infants in front of a monitor with the camera attached. Once the observers had the infant's attention, they would begin to show the infant a series of repeated and non-repeated images until the child met the researchers' designated criteria. Data was collected based on the number of times the infant looked at the image, as well as through heart rate and event-related potentials. After observing and testing 45 infants, ranging from 4.5 to 7.5 months old, the researchers found that the infants in the 7.5-month-old range showed reduced amplification of neural correlates when presented with repeated stimulus during late repeated trials. These results seem to point to older children having greater processing efficiency and object recognition. So what does this mean for the realm of object recognition? This particular study is significant to the field of object recognition because it provides evidence of the many mechanisms that take place in order for object recognition to occur. The researchers found that variables such as age, attention, stimulus type, and stimulus repetition all play a role in the infant's development of object recognition. They also recognize that other internal and external processes, such as arousal and social experience, play a role. Further studies of these variables could in turn give researchers further insight into the field of object recognition and behavioral cognitive psychology as a whole.
Prosopagnosia can be described as the inability to recognize phases and can occur from brain damage or as a developmental variant. Developmental prosopagnosia is much more common than acquired prosopagnosia. The neural basis of prosopagnosia is still unclear, but there are two main schools of thought. The first is that prosopagnosia can be marked by altercations in various regions of the face network, specifically the fusiform gyrus. The second view suggests that there is a disconnection between the posterior and anterior regions within the face network. There is currently no treatment for prosopagnosia. Developmental prosopagnosia is a lifelong disorder, and spontaneous resolution of acquired prosopagnosia is very rare. However, there is hope because it is unlikely that acquired prosopagnosia affects all areas of the facial region network.
Many researchers and clinicians believe that face processing deficits in prosopagnosia cannot be improved. However, evidence for effective treatment for prosopagnosia has just recently begun to come to light. The current literature elaborates upon treatment attempts in both acquired and developmental prosopagnosia. To begin, it should be noted that those with acquired prosopagnosia can sometimes undergo spontaneous recovery of facial processing systems after brain damage. This is rare, however, which is why researchers aim to develop compensatory treatment approaches. These treatments seek to teach patients ways to adapt to their face recognition deficits by using the systems that are intact in the face processing systems. Compensatory training has shown more potential for treatment approaches in those with developmental prosopagnosia. Researchers also focused on remedial training, which targets the underlying deficits to promote normal face processing. Again, this has shown more improvement in those with developmental prosopagnosia. Another approach studied was the administration of oxytocin. Oxytocin has shown to be involved in aspects of social cognition. It has been shown to be involved in tasks that require sensitivity to subtle information from the eye region. This is relevant to prosopagnosia because the eye region is used to identify faces. Patients that were given oxytocin performed better on a face memory task than those who were given a placebo. In conclusion, even though the current treatment studies are promising, more research needs to be done to create long-lasting treatments in patients with developmental and acquired prosopagnosia.